This video will cover the countercurrent mechanism, which is basically the way by which the body can concentrate urine in case you're dehydrated. And so this is a pretty detailed explanation of this mechanism. And the very first thing that we'll need to do is talk about what a countercurrent actually is. So you're going to see this means that fluid flows in opposite directions through adjacent segments of the same tube, both before and after a hairpin turn. And this is going to happen in two different places around juxtamedullary nephrons. So one is the loops of Henle. These are a countercurrent multiplier. So if you look at this on the way down and on the way back up, there's going to be flow in opposite directions, and this is the countercurrent. Now this is a countercurrent multiplier, and I'll explain what that means a little bit later on in the video. The other structure that has a countercurrent here is the vasa recta. And so the vasa recta, its flow of blood, again, is a countercurrent, as you can see. And it is a countercurrent exchanger. And towards the end of this video, I'll explain the difference between a multiplier and an exchanger. The countercurrent multiplier is the mechanism by which the kidneys will produce concentrated urine in a small volume. And this happens especially when the blood plasma concentration of antidiuretic hormone is high. Before moving on, we'll need to introduce a little bit of terminology here. A hypoosmotic solution occurs when an area, cell, or space has less solutes than another. Water will move away from a hypoosmotic area towards the other area or cell or space. With isoosmotic, you've got two different areas, cells, or spaces with approximately the same amount of solutes, and there's no net movement of water toward one or the other area, cell, or space. Hyperosmotic means that an area, cell, or space has more solutes than another area, cell, or space, and so water is going to move toward this area and away from the other. Urine is concentrated, meaning hyperosmotic, relative to blood plasma. And this is one of the reasons why we can survive with limited amounts of water. So every day we have what's called obligatory water loss. And this is the water that it takes in order to dissolve and excrete the urea, sulfate, phosphate, and other wastes that we need to secrete. We require to excrete at least 0.444 liters per day. Now, given that we absolutely have to excrete this amount of water per day, if you have inadequate water intake because of this obligatory water loss, this is definitely going to result in dehydration. The hyperosmotic interstitial fluid in the medulla of the kidney is your actual key to the countercurrent multiplier system. And this hyperosmotic interstitial fluid in the medulla is actually set up by the Henle's loop of the juxtamedullary nephrons, which again you can see right here. Now it's the opposing flow in the two limbs of the loop of Henle, descending, which you'll see on the left, and ascending, which you'll see on the right. This again is a countercurrent flow, as you can see by the arrows that are within these limbs. Now the entire loop is a countercurrent multiplier system, and what this is going to do is to create a hyperosmotic medullary interstitial fluid. So we're not really talking about the fluid within the descending and the ascending limb. We're talking about the fluid that actually surrounds them. And so there are two keys to this. First of all, that in the descending limb, you have H2O, which is able to leave by osmosis. And in the ascending limb, you have sodium chloride, which is able to leave via active transport. That's why those little circles with the crosses in the mean. So there's a different mechanism for how H2O leaves the filtrate on the descending limb versus how sodium chloride is reabsorbed from the ascending limb. Now, at the end of the proximal convoluted tubule, the fluid that enters the loop of Henle, is actually isoosmotic with blood plasma. And this is isoosmotic at 300 milliosmoles per liter. So you can see that both at the beginning and at the end of the proximal proximal convoluted tubule, you have the same osmolarity. And again, the reason why, I think we mentioned this in the previous video, is because in the proximal convoluted tubule, both water and solutes are reabsorbed in the same proportions, which is going to keep your osmolarity the same. Now, what happens in the descending limb of the loop of Henle where the water leaves, this can only be understood in the context of what the ascending limb is doing. So in the ascending limb on the right, you're going to see sodium and chloride reabsorbed and moving into the interstitial fluid along this entire length. Now, the reason why you don't see water moving along with this solute is because the ascending limb is actually impermeable to water. If you'll look at the diagram itself, one of the things that I particularly like about this diagram is look at the border of the ascending limb. You'll see that the border is this thick brown border, and that indicates the impermeability to water. Now, sodium chloride leaving as you go up via active transport means that you're going to lose sodium chloride all the way up the ascending limb. So this is going on. You're seeing the sodium chloride leaving the ascending limb. Now, because there's a counter current going on, at the same time that you've got this ascending filtrate in the ascending loop, the descending loop itself is highly permeable to water. And so water is going to leave by osmosis. So you're going to see water leaving all the way down the descending limb because, as I said, H2O leaves the tubule by osmosis. So as you move down and then back up, the osmolarities of the filtrate inside the ascending limb and the interstitium are equal to one another. One thing that I want to point out is that there's going to be probably a little less water leaving at the bottom part of the descending limb because there's less water left. It's been moving out all the way down the descending limb. Now, as you come back around to the ascending limb, as those active transport pumps really get cranked up, basically what this is going to do is as the sodium chloride first leaves, you're going to be able to move a lot of the sodium chloride out with those active transport pumps. As you continue to move up the ascending limb, there's less and less sodium chloride in the filtrate. So basically it means that up at the top of the ascending limb, because there's less sodium chloride in the ascending limb, you're going to be moving less of it out with those active transport pumps. And this is the basic reason why sodium chloride is more concentrated in the inner medulla than it is in the outer medulla. So the upshot of all of this is that as you descend down into the medulla, it becomes more and more hyperosmotic. So basically you can think of the interior of the medulla as a lot more salty than the outer portion of the medulla that borders the cortex. So again, taking a look at kind of the big picture here, the ascending limb is what I want you to focus on first. It's impermeable to water. It's permeable to sodium chloride. You've got active transport pumps pushing sodium chloride out from the filtrate as it moves up the ascending limb. Now again, it's more concentrated at the bottom here. So you're going to be able to move out more sodium chloride through those active transport pumps simply because there's more sodium chloride to move, if that makes sense. Now, as you ascend through the ascending loop, there's less sodium chloride in the solution, therefore less sodium chloride to push out, therefore less solutes in the upper portion of the medulla. So it's the ascending limb that kind of sets the osmolarity of the interstitial fluid. And again, what that means in the descending limb is that you're going to see H2O leaving all the way down the descending limb, but you're probably going to be seeing a little bit more of it leave at the beginning because there's simply more H2O in the solution to leave. Now, one last thing that I want to point out is in the proximal convoluted tubule, you saw that this was isoosmotic to blood plasma. Well, in the distal convoluted tubule, as you leave the ascending limb, you're actually hypoosmotic. So look at that, 300 and then 100 and then 100. So your osmolarity of your filtrate is actually going to be less than it is in blood plasma. So this is very dilute stuff right now. So your combination of the ascending and descending loops of Henle, this results in an ion gradient. And so deeper portions of the medulla become increasingly hyperosmotic. And again, this hyperosmolarity will concentrate the fluid that gets left in the tubules. Now, I want to make this clear that flow through the loop doesn't stop while these things are moving out of the loop. And what this results in is an osmolarity difference at each horizontal level of about 200 milliosmoles per liter. Now, that's not reflected exactly in the diagram on the right, but what it does mean is that the difference gets multiplied. So your osmolarity difference gets bigger and bigger as you move deeper into the medulla. That's the reason why this gets called a countercurrent multiplier. By the time that the fluid reaches the bend in the loop, if you look at the bottom there, the osmolarity of both the tubular fluid and the interstitium has been multiplied to a very high osmolarity, which is going to reach about 1200 to 1400 milliosmoles per liter. And you can see that right there. Now, as the fluid moves back up the ascending limb, the osmolarity decreases because of all that sodium chloride that's leaving. And as it turns out, as I mentioned before, the fluid that enters the distal convoluted tubule is actually hypoosmotic compared to the blood plasma, and it's going to be at about 100 milliosmoles per liter. Now, during passage through the distal convoluted tubule, you have still more sodium and chloride moving out of the tubule into the interstitial space, but it is still impermeable to water. And so it means that your osmolarity is going to drop even lower. It's not represented on the picture here, but by the time you reach the end of the distal convoluted tubule, it can be as low as 80 milliosmoles per liter. Now, what happens at this point? Because at this point, you're going from the nephron into collecting ducts. And so the collecting ducts are actually going to be what's going to determine whether you're going to excrete this dilute fluid or whether you're going to concentrate it. So if we look at this, if you have antidiuretic hormone present, then water reabsorption is going to occur in the collecting ducts. And so what I want you to notice is that H2O is going to leave. It's going to continue to leave as you move deeper into the medulla because of the gradient that the loop of Henle actually set up. So even though the filtrate in the loop of Henle changed on the way down and changed on the way back up, where does this become really important? It becomes really important in the collecting duct because the gradient that your loop of Henle set up is going to allow that much more H2O to leave if you have ADH present. So if ADH is present, it's going to open channels in the collecting duct called aquaporins. And these aquaporins are going to allow H2O to diffuse out into the interstitial fluid. Now, if you had not set that gradient up with the loop of Henle, then not a lot of H2O would diffuse out. But because you've set that gradient up, the deeper that you go into the medulla, water still continues to leave. And what you end up with, as you can see at the very bottom of the collecting duct here, is a small volume of highly concentrated urine. So this happens when you have antidiuretic hormone, meaning that you're not going to urinate nearly as much. So as you can see here, water continues to move out of the tubules throughout the length of the collecting duct as you move from cortex to medulla. And again, as I said in the previous slide, as you reach this deeper portion of the medulla, you're going to see where it says 1200 at the bottom of the collecting duct. This sort of mimics the 1200 at the bend of the loop of Henle. So basically, because water continues to move out of the collecting duct as you move deeper and deeper, This is going to allow you to extract more and more water as you move deeper and deeper, and so you're going to have smaller amounts of concentrated urine in an attempt to try to prevent dehydration. Now, what is it that happens if you don't have ADH present? Well, if you don't have ADH present, then those aquaporins don't open up in the collecting duct, and therefore the collecting duct is impermeable to water, and that highly dilute urine passes on through and gets excreted. And so essentially what's happening here with the countercurrent multiplier is that the loop of Henle sets up a gradient of solutes that gets higher and higher the deeper that you go into the medulla. And this is what the loop of Henle sets up just in case you need to reabsorb water if you're getting dehydrated. So you can think of the countercurrent multiplier as sort of dehydration insurance. You're not going to have to use it all of the time, but that gradient will be there to extract water if you need it. And so if you don't need to extract water, if you're not dehydrated and you don't have ADH present, then you're going to excrete hypoosmotic urine, a large amount of dilute urine. Now, in terms of the vasa recta, this is that long blood vessel that follows the loop of Henle both down and up, and this was called a countercurrent exchanger earlier on. So in the vasa recta, in the descending limb, you're going to see sodium and chloride diffusing into the vasa recta in the descending limb. And again, that's going to be on the left here. You can see that sodium and chloride are going out as you go deeper, and that are coming in, and water is going to be leaving as you go deeper and deeper. Okay, as you go back up the ascending limb, this gets reversed, and you can see again as you go back up the ascending limb, water will actually enter the vasa recta, and sodium and chloride will actually diffuse out of it. Okay, and what's your result here? Your result here is that the vasa recta doesn't affect this gradient, and that leaves the countercurrent multiplier system intact. So as you'll see in the blue writing below the blood vessel on the diagram, the vasa recta is highly permeable to both H2O and to solute. It's also got a really sluggish blood flow through it, and that means that the blood in the vasa recta is going to mimic the osmolarity of the interstitial fluid that surrounds it. This means that the blood becomes more concentrated as you descend deeper into the medulla, but when you come back up, it gets less concentrated as you approach the cortex. So basically, why mention this? It's because if the vasa recta was impermeable to either H2O or to the solute, it could potentially affect this countercurrent multiplier on its trip through the cortex. So the fact that the vasa recta does nothing is because it is a countercurrent exchanger, not a countercurrent multiplier. So it exchanges both water and solutes at an equal rate to what's in the interstitial fluid around it. So here's our sort of side-by-side comparison. So again, with the loop of Henle, you have a descending limb that is permeable to water, not to solutes. You have an ascending limb that is impermeable to water, permeable to solutes. So you have different things leaving at different times that sets up that osmolarity gradient. Now the vasa recta on the right side, again, it's highly permeable to both H2O and solute, so the blood that's inside the vasa recta will change on the way down, it'll change on the way back up in proportion to the interstitial fluid surrounding it. Now there's one more thing that we've got to consider that we haven't talked about much, and that is urea. And so this is the main means by which we excrete nitrogenous wastes. And so urea is actually also going to contribute to this hyperosmotic interstitium that's required for our countercurrent multiplier system. So I mentioned sodium chloride coming sort of out of the ascending loop and water coming out of the descending loop, these things setting up our countercurrent multiplier. So what effect does urea have on this? Well, first of all, urea is freely filtered at the glomerulus, so it's going to leave the bloodstream very easily. You can see that it's a relatively small chemical, so it's going to leave the glomerulus very easily. Now, 50% of urea actually gets reabsorbed in the proximal convoluted tubule, with the remaining 50% entering the loop of Henle. Now, we're going to take a look at this on the way, all the way through here. So in the loop in the collecting ducts, urea gets reabsorbed, secreted into the tubules, reabsorbed again. Some urea gets trapped in the interstitium. But let's take a look at our slide here and just follow this all the way through. So at the proximal convoluted tubule, you saw that 50% of the urea that is in the filtrate gets reabsorbed, leaving 50% in the fluid that enters the loop of Henle. So in the loop of Henle, at the very bottom of the loop of Henle, you get some absorption of urea into the filtrate. So you're going to get 50% that's brought back in, leaving your urea concentration at 100% of what it originally was in the filtrate. So you continue on around the distal convoluted tubule, and in the cortex, the renal cortex, you're going to get 30% reabsorption into the interstitial fluid surrounding the tubule, which is going to leave you with 70% of the urea that was originally filtered. Now, once you get down into the inner medulla, you're going to see something else happening, and that's going to be 55% of the original urea that gets reabsorbed into the interstitial fluid. So your 70% minus your 55% means that 15% of what you originally filtered out of the blood plasma is actually going to leave in the urine. And why would this happen? It's because this 55 reabsorption and the 50% facilitated diffusion that kind of cycles through the tubule here is going to help maintain that hyperosmolarity that you need in case of dehydration. So again, some urea gets trapped in the interstitium, contributes to the hyperosmotic nature of the interstitium, and so basically this is how your countercurrent multiplier actually works.